Okay, so so what we discussed at the last point of the previous lecture was the fact that actually we agree that we have all the genetic material required to be enclosed in a vesicle, and then there's a jump from that point to the synthesis of the first cell that was alive and had defined life as we know it, which evolved over a period of billions of years and finally gave us this biodiversity that we are experiencing today, currently. Okay, then the question then arises, that the genetic code that we see, which is there existing in the cell, this is there in all biological systems. In each and every biological system that is there, the genetic code is present, right? And so now the thing is that if that is true, then what is it that is so special about this code, and how is it that this code gradually evolved to give us the biodiversity that we have? So that question is partly answered by research, and there's continuing research in this. And we talked about a few things in the previous lecture, where we discussed, for example, that these codes, the coding can have an error when you translate from a genetic code to a protein, or when the gene itself is being replicated, you can have an error, and that error is the frequency that it occurs in is 1 in a million, and so it's called a 1 in a million code, standard code, and we find that if we really wanted to change that code, then it's not so easy to do it because already the entire evolution has supported this code, and any change would essentially be a disruption in the process uh, of uh, of actually converting the information to the protein from the DNA to the proteins and so forth. So that would essentially cause a disruption. Now, we need to think of the principles of evolution that actually essentially shape this universe, and and if and perhaps this would be an underlying principle that would tell us how life would form on other planets. If and we discussed a little bit about that in the previous lecture. All right. Now, so let us look at a few facts about. Let us look at a few facts about what we have as information with us right now. So if I were to ask you how much DNA does an organism have, well, we can't. It's not really a general. You can't really have a general answer to this. So you have to go organism by organism. And for example, if we talked about E. coli, then we could say that it has about in the range of about 5 million bases. If we talked about Homo sapiens, then we have a trillion bases, let's say. And then if we talked about the copying mechanism or base pairing, then that would be a special feature that defines life as in general. So how it does and what it does, those are things that we have to go back and try to understand in the context of how evolution has shaped this. But the question of life in general, and if we just look at E. coli and take that say as a simple organism, then can we ask the question, how much DNA does an organism need to have to function? So this again, is a question for Nitish. Maybe can you comment on this, Nitish? Anybody? I don't know.
Sir, it has as much, at least DNA, so that it can have genes to replicate and produce a few proteins which is responsible, which is necessary for replication.
So that's where the difficulty is, right? So what do you mean by that an organism needs to function? So this question is again as something that I left as an open-ended question. So you need to go back and look at some more literature to really come up with what is the current status of answer. for this question, because there is no complete answer for this question yet, because this is an ongoing research where people are trying to understand, if I were to construct a minimalistic cell, then what are the things that I need to put in this cell so that it essentially functions as a living organism without having the paraphernalia of all the other things that is required. Let's say the extra redundancies which are there, can I get rid of those and still it functions? Because we have a lot of, in all of these genes, we have a lot of junk DNA, we have a lot of non-coding DNA. So then how are those things complementing an organism, or is it completely useless? So that kind of question is still not answered, because time and again people have found that actually some things that were considered to be not important are now finding new reasons to say that they are very important. So think about these things. And so I'm leaving this question as an open-ended question that we can perhaps revisit later on. Okay. Now, the whole idea of this cell being able to reproduce the information that it has and produce another cell exactly like itself is based on the genetic code that is embedded. And we need to understand how this genetic code got deciphered. So this is a little bit of a historical thing, but even then, we can actually go back and look at what's really happening here, and try to kind of retrace the steps to the formation of the discovery of the structure of the double helix, and then the formation of the analysis and the discovery of the genetic code. So we know that if you talked about gene as a unit of inheritance, then you have to go to Mendel, and all of you know the work that was done on these green peas which were there, and how based on the variety of green peas and the type of green peas that were being formed, he had done a series of documentation, and then he talked about inheritance. And that work was rediscovered in the 1900s after 35 years. But at that point also, the nature of the gene was still not known. And it was in 1944 that somebody had established that the gene is made up of DNA, right? Then that gave the kickstart to the new theme for doing the research for trying to find out what the DNA or what the gene or the genetic inheritance code is. And keep in mind that this is time of the World War, 1944. And so during this time, besides this also, a lot of work was also done in terms of, finding out what the neuron looked like. And if you go back and look, results, the scientists were working even under those really adverse conditions and made some amazing discoveries during this period of time. Now, 1953, Watson and Crick, of course, they discovered the structure of the DNA. And how many of you know that one of the other persons who was also contending for this was Linus Pauling?
Yes, sir.
Yeah, okay. So you know that, right? So that story that, he could have actually claimed it because he had done a substantial amount of work, but he just let these people take it forward after a discussion. Else he would have been contending for the Nobel Prize also, the third Nobel Prize, I think. So anyway, so we do know now that the DNA has these 4 bases: adenine, cytosine, guanine, and thymine. And the RNA is AGCU, U being the uracil, which is replacing the T from the previous group. And we know that the double-handed strand looks in the order of this kind, where we have a 5' to 3', and the reverse on the other strand, 3' to 5'. And you can see that they will be bonded, and some of them will have 3 bonds, and some of them will have 2 bonds, right? Which have 3 bonds and which have 2 bonds?
GC would have 3 bonds and
Yeah.
AT would have 2.
Correct, 2 bonds, correct. So we know that now. And so this was all fine, but then the big question remained, and it was still unanswered. It is that how is this information that we have, which is on the DNA strand, getting translated to protein? So obviously there must be that this must be the basic alphabets of life, but we don't know how this alphabet is getting translated to the language of life, right? So that's what happened then. And at that point of time, this person here, George Gamow, constructed what, or rather created what was called the RNA tie club. How many of you know about this? The RNA tie club? No? All right. So, so what he did was, he was a physicist, and he was already very famous for his uh, propositions of the Big Bang theory and the formation of stars. He was very well known for that. And so he made a what's called a brotherhood of 20 regular members, so that each of the amino acids, and 4 honorary members for each nucleotide. And 8 members of these were already Nobel laureates, or became Nobel laureates later on. One person became Nobel laureate later on. So it was a very high flung and elite club who were trying to essentially, so they had the best minds trying to discover the genetic code, how it was being translated. And so they were doing all this work, and some of the ideas at that point of time was proposed. One was being spearheaded by Gamow, and the other one was by Francis Crick. So Francis Crick had this adapter hypothesis, which he said that there must be some unknown entity which actually essentially helps in translating the nucleotide sequence to the amino acid sequence. And Gamow then proposed that actually, based on his analysis of just looking at the 20 amino acids and the 4 bases, he came up with the idea that the codon should be a 3-letter code. And of course, the codon word was not used then, but a 3-letter code would be sufficient to define all the 20 amino acids. So these are the 3 things that he started with going out with. And Gamow made this proposition that if you had the top and bottom being identical, like A and A, T and T, then CG could be having 2 orientations. So based on those 2 orientations multiplied by 2, and you could choose any one of this pair by 4. One of this position, because if one is fixed, the other one is already fixed. Then 4C1. And if you had if the number of diamonds at the top and bottom were different, then it becomes 4C2, and it's all options. So that gives it those 20 amino acids that he was looking at, and so he thought that he had this whole thing in his bag. But there were other propositions, and one of them is being made by Francis Crick himself. And but he made some incorrect conjectures, but came to the right result. So that goes to show that during those documentations, whatever was documented, that you could start off wrong, and that's not a problem. I mean, you can always learn as you proceed with the research. So he had said it like this, that this is, and you can see this 4 to the 2 and 4 to the 3 kind of logic. So there must be redundancies. So he was almost certain that there would be redundancies there. And of course, they knew, because they had the structure, that there would not be any comma. What is the meaning of comma? That there would not be an intervening compound or an intervening molecule which would separate out the ATGC, and then so there would be 3 codons, 3 bases, and then something that separates it out, and then another one. It would not be like that, because they knew already from the DNA structure that it was continuous. And so this one was very correct, what he said, that there would be only one reading frame. That means if you start from one point on the sequence, then you could group it in one way. If you started with A, for example, or you could group it in another way if you started with B, or group it in another way if you started with C. But only one of them would be true. Not all of them would be true at the same time, which was correct. But he made an incorrect conjecture here, where he said that that these repetitions would not be allowed, because the reading frame could not be defined in this case. So that was not correct. Then the other thing was that if you leave these 4 out from the 64, then you get 60. So that should be the main codon, but that was also not quite correct because he gives another example that divided by 3, that should be 20 amino acids. So although you come to the right result, the logic that was followed was not quite correct. And we see how that was happening. So this person was relatively unknown at that point of time. So he had joined the National Institute of Health. So Dr. Marshall Nirenberg had joined there, and he was a young fellow at that point of time. And he was also trying to understand this problem. And so there was this conference, I forget the location, where all these big people were there trying to present their recent updates on their work on the genetic code. And he just had some preliminary results. And so he had gone there. So what he had found in his lab was that, he had done a set of experiments where he used what are called the cell-free extracts. That means you break down a cell, take out the extract of the cell, so that you know that all the components that are required for the cellular machinery work are already there. And then you treat it with a specific kind of experiment to see what the result is. Because at that point of time, not much was known about, what are the elements. Some things were known, for sure. Like they knew that the existence, by then they knew that there is something called the tRNA. They knew also, how to synthesize sequences of the RNAs and or the nucleotides. So all of that was known. So they knew what ribosomes were, and that these were instrumental in actually translating the RNAs into proteins. Not completely, but some of it. How that they knew that this was required, but exactly how certain mechanisms and other things still were not completely known. So what he did was he essentially used a poly-U sequence, and with the cell-free extract and he used 20 different test tubes having labeled amino acids, and he found out that the one in which you had phenylalanine that produced a sequence, and so he knew that the UUU was coding for phenylalanine. So that is the work that he presented there. And initially in his first lecture, there was nobody there present. A few people were here and there, but it so happened that Francis Crick was there. And when he heard that this had happened, he knew that this person had cracked the code. And so he told him to make a big presentation the next day again. And in this time, they had the entire audience almost of the entire conference was present there. And then he made this presentation of what he had done. And of course, later on, this whole work got refined, and you also had along with him, you had Robert Hawley and Hargobind Khurana, who also got the Nobel Prize along with him. Hargobind Khurana got it because he was able to come up with methods that could precisely synthesize chains of the mRNA and exactly where you could link it. So you could get these chain lengths, of certain sizes, and exactly what kind of repeats you want. So he was able to do that so that his method was used to synthesize variations of the genetic code. And Hawley was a person who discovered the tRNAs. So together, three of them, as you can see, were able to come up with a decipher of the genetic code. So the method was followed is like this: that you have a filter which will allow the trinucleotides of this kind, tRNAs pass through, and whereas ribosomes would not pass through. Now, whenever the reaction takes place, it was known that you have the tRNAs, the ribosomes, and the sequence length that would be present. So Nirenberg was also trying to answer what is the minimum sequence length that is required for this to attach to this, for the tRNA to attach. And so that would also tell you what the code length was, right? So and this is what he did. So when he did the experiment, he found that that this would be retained on the surface, and because this phenylalanine which was there, which was labeled with a C13 or a C4, I'm not sure, one of the two. It was labeled with a radioactive carbon. So based on the analysis, he could find out that this was phenylalanine. And to kind of eliminate the fact that there could be any other tRNAs. So they also added, for example, the tRNA for arginine and leucine, and found that they passed through, but not the one that was bound to the ribosome. So that kind of give him the complete, uh, uh, give him the ability to completely decipher this entire code, genetic code. So now with him, as you can see, he had there was also his lab mate Johan, who also helped him out, and so together, along with the other Nobel laureates, they were able to decipher the entire genetic code. So what it goes to tell us is that even if you have an elite company of Nobel laureates, 8 Nobel laureates in one group, it just requires somebody completely unknown who can actually break the code, right? So this is the code now, as you can see. So it's nothing but a mapping of codons, which is set C, to the set of amino acids 20 in number. And you can see that the first letter of the codon is on this column here. The second letter is here. And the third letter is here on this side. So U, U, and U, which is on this side. U gives you phenylalanine. UUC also gives you phenylalanine. So there's a redundancy of 2 in here. Look at leucine here. So leucine has CUU. So this is starting as C, then U, U, and then C. So CUC, CUA, CUG. Then now there's serine here, and there's a serine here. So you can see there are 6 of these which are required, or rather 6 of these which are coding for serine. So serine has a redundancy of 6. So instead of 1, it has 6 of these which are there. So likewise, you can go down the list, but you'll find that methionine has 1 only, and there are some which have only 1. So this methionine is, where is that gone? I can't find it. Yeah, there. So AUG. So that's methionine. Starting. Huh? Can you hear? Can you hear? So AUG is methionine. So that is the start codon. And then you have the stop codon. Can you hear me now?
Yes, sir.
Any
Yes, sir,
question?
you're audible.
Yeah, question? Somebody was asking something.
No, sir.
Okay, all right. Fine. So now I just want to go briefly into 2 aspects of this. Is why would we say that this genetic code is good, and is there some lead about how it all evolved? So I'll just spend some time on that. So if you were to group it on the mapping that I've shown here, so this vertical one is the relative amounts of the sizes of the amino acid here, and this is the hydrophobicity on the x-axis. And if you were to plot them, then you'll find that you group it together according to the columns. So this is column 1, column 2, and column 3 here. But column 4, as you can see, is a little scattered. So there are only 3 in column 4, and they're a little scattered here. 1, 2, 3. I think there are 3. No, 4 here. There. 4th one is here. So glycine is of course, as you all know, it's the most flexible of the amino acids, and so its position here is not surprising. But the others are structurally related. So obviously there's less structural specificity as you come to the 3rd column, or rather 4th column here. Okay, so the fourth letter that comes into each of this. Sorry, 4th column that comes in, not letters. So 4th column of this with the letters that are for each alphabet that is there. All right. So now the question is there some clue as to know how this coding could have started in the first place? Obviously, such a sophisticated thing would not have happened right away, And so there are a few other questions that are there. So let's see if we can answer those questions. Okay, the question that I have is, I've given some things here, but I still have one question for you. Maybe somebody can answer. Is let's see if somebody can think about this. Do you think that it would have been, that you would have, one codon, one alphabet at a time assigned to one of the amino acids, and would the tRNA have been synthesized right from the beginning or later on? So there are two questions. The first question is, do you think that the way the codon would have evolved is that it would have just evolved as first as a single codon, single alphabet, and then there would have been two alphabets and then three alphabets. Do you think it's like that, or three at the same time? That's the first question. And the second question is, do you think that the tRNA would have co-evolved, or do you think that it would have evolved later and separately? Yes? Somebody can answer that question. Somebody would like to participate in this? Otherwise,
So could you repeat the question once?
Yeah, the first question is that do you think the codon would have evolved with one letter first, and then the second letter, then the third letter? Or do you think it would have been three letters at the same time, right from the beginning? And the second one is, do you think that the tRNA would have co-evolved with the genetic code right from the beginning, or it would have been a later addition?
Sir, it's in case of like one letter at a time, we must have like in case of one letter at a time, or right from the beginning three letter at a time, we must have the tRNA according to them. Like in case of one, if it is reading one letter, then there must be corresponding tRNA for that. Like if there is A, then corresponding to A, there must be tRNA. Then there must be tRNA which reads
Correct.
two letters at a time.
I agree with you, what you're saying. But the question then is that when you combine these two questions, do you think that you would have three codes at the same time, or one code at a time that evolved?
I guess having one code at a time would be problematic, because then how would you actually charge the tRNA with so many different amino acids, having only like 4 different tRNAs?
Correct. So actually it's going to be very difficult for evolution to redo the entire thing. So it would have co-evolved so that you had three codes at the same time, with a tRNA that evolved along with it, with three codes that it could read, three letters that it could read of the code, right? So that is important. So good that you at least are able to kind of infer that. So you see, there are people waiting in the lobby. Who are these people waiting in the lobby? I don't know who this is. Chail Singh, I don't know who this is. Anyway, I'm letting him in. So you see, what they're showing you is that what I'm showing you here is a table where we are looking at the first probable codons for, let's say, valine, alanine, asparagine, and glycine. Now, this is taken from the fact that there are 10 amino acids, and these are the first 10 amino acids that are shown here, that have been always shown and agreed upon to have the highest abundance right from the beginning. So there are various kinds of evidences that are there in the literature, plus the experiments of Miller and Urey also showed that the highest concentrations and abundance were for these amino acids. And that's quite surprising, because what we find is that out of this, the top 4 actually are right here. 5 means I don't see, there could have been another one which I kind of missed out. Even valine is there, so glycine, valine, alanine. So glutamine is not showing up here, but maybe it can come up. So now you see what the early version of the code would have been, that you start with a G for one of this, and then you don't care what the other two codons are. It doesn't matter. It could be anything, but it'll still select one of these from there, right? So that's the way. So maybe that was the early code version of the code would have been, that would have been a G in the first place, and maybe one would have been specific for the others, and the third one would not have been mattering so much. So this is the way it could have perhaps evolved for the first column, second column onwards. So you can see the logic that flows here. That given that you fix one of this, you could choose this one just by one, and you wouldn't care what the other one was for that. Or you choose this for this, and you wouldn't care what the other one was for the other one. So what the evidence shows is that it would have started with a G in this case in the early, and then it would have actually given you these codons which are actually going to be constructed for this. All right. So there's a whole paper on this, and I can share that paper with you later on. Okay. So anyway, so this is how it later on got rearranged so that you could essentially have the early 10 amino acids accommodated in this whole structure. So you see the valine. So you can see the valine, alanine, asparagine, and glutamine with the glycine, the 5 bottom, the G. This would have been the G codon, right? See there. So that part you can see easily what that relates to the previous slide. So, and also most likely the earliest RNAs, tRNAs also had an anticodon loop just like this one, and had similar structures, and it essentially co-evolved with this. Because as we know that whatever is the mechanism, it would have been a huge shift to try to reinvent the entire mechanism by which ribosome shifts along the mRNA, and the tRNA comes in and then joins, and then gives you the addition of one amino acid along the chain. So all of that would have required a huge amount of evolutionary energy, if you may put it that way, and that could have not been redone. Once process is in place, it should have continued. So we can see that this is what have happened. Okay. All right. And so if you wanted to make any change, like I said earlier also, then so you can see this is the penalty that would have been given for a change. So the penalty should reduce. So you can see if you had threonine in the first place, and you wanted to shift the threonine to some intermediate position, and if you give it a, so according to this more the problem that these people are doing, they give it an intermediate shift so that it would change this position and then try to minimize it. And they see that there is a new position for this threonine possible. But if you were to change threonine from column 1 threonine in column 2, and then it would have been it would have been difficult to move this out. So adding something to column 1 would have been made. So it is favorable to add leucine in column 1, threonine in column 2, and glutamine in column 4, as you can see if those changes were made. So you can see that if threonine is added to column 2, leucine in column 1, so this circular is there. So the circle shows you that circle would have gone down, but the threonine goes through a higher intermediate, so that doesn't work out for here. Whereas threonine goes through a lower intermediate where it goes down there, so that would have been favorable. Then here we see also there is a line that goes down this way, so it would have been easier to join this one. And the fourth one is right here, so that would have been the easiest. So sorry. Here we see that the barrier is there, and obviously for arginine, because the barrier occurs here, this cannot go down further here. So it has to go through that barrier, and that is not energetically favorable. So if the penalty is higher, then it can't go through. So there is a selection barrier or change for any change that happens in the genetic code. So now what I'll do is we'll have to kind of recap and then go to the next level. So what have we learned so far? Can we have a short discussion on this? Can somebody kind of summarize this for me? You have to participate, otherwise, what will happen is it'll all be one way.
basically what we saw today was
Yeah,
like
just,
how.
Tell me.
Sorry, sir.
Continue.
Yeah. So basically what we saw today was like the principles of how the genetic code actually evolves. Like we can see that earlier probably it wasn't as complex as it was as it is today, and like like in the starting we saw how people actually found out what like basically the how DNA goes to RNA and RNA goes to protein, and how the ribosome actually translates that information in the mRNA, and and how basically basically how people found out that it is triplet codons that actually code for the genetic information sequentially, and we looked at what were the possible mechanisms that which were followed in the basic evolution. That is, how did the codons actually come into being?
Okay, Ratnesh, would you like to add to this?
Not much, sir. Like this is what we have done today.
Okay, no, but so overall, let's look at this overall one more time, so that we kind of get a perspective, kind of consolidated. So what we did was we see. So if we look at what we had done earlier, we had looked at systems biology, and we wanted to kind of just see why this field of system biology evolved first of all, and we talked about the fact that, well, because the cells have become, cells are complex in nature, and over a period of time, the need to understand how cellular functions were taking place, both commercially for commercial reasons as well as for reasons of trying to address health problems, to understand disease and then mechanisms. People realize that you have to take the help of some kind of analytical methods to take care of these things, and that also became very important because you also had newer instrumentations and newer methods of data analysis, newer methods of experimentation coming up, which were high throughput experiments, right? So you were getting a lot of data, and there was no methods to analyze it. So that's the reason why systems biology evolved, because it was giving the bridge between engineers and other areas of science, engineering science, with respect to what biology was saying, and to help biology to grow faster. That's the reason why it evolved. But then it leaves that question, if cells are so complex, how did it evolve? And so the question, that parallel question of, how did life come into this planet to give us a biological world, is required, because unless we understand how life evolved, and unless we understand the fundamental genetic code and how that evolved, we cannot take this forward to understand the methods that we would be applying, or when we write code or when we write equations, we would not have a understanding of the physical aspects of the problem. I mean, what is really happening in real life, what is happening within the cell, that understanding is required so that you can do a better job in terms of analyzing data that comes your way. So that was very important. So to understand that, we went back to the basic question of how life evolved, and what we did was we looked at three basic methods by which organization takes place, because it was very clear that you have to have in that chaotic environment some kind of self-organizing principles. And what we showed, that just by the physical laws that are present, just by the chemical laws that are present, and just by the presence of these amino acids and the primordial soup, whatever was there, just that was sufficient to organize them into groups that could be useful, or doing something that was different from just being present in a primordial soup, right? So one of the things was to understand how this physical organization takes place, and we showed that it can happen very easily. One of the examples was showing, the convective currents. The convective currents showed you that just by one change of one parameter, you could get organization. So therefore, locally, somewhere, some condition would be there which would fulfill so that you could get organizations in terms of physical conditions. Similarly, for chemical conditions, you could show that you could have reactions that could actually have a periodicity, which we saw through those Belousov-Zhavitinsky reactions, right? Similarly, we saw that, lipid bilayers could form. Anything that was, amphiphilic, any molecule that was amphiphilic would in certain contexts give you organized shapes. And those organized shapes were very well defined. And there are equations that can make you, or rather equations that you could derive, that you could use to construct very specific kinds of molecules and shapes, right? Using very specific kinds of molecules, you could create very special shapes, and one of the shapes could be the vesicular shapes with a lipid bilayer. So therefore, that also was something that was possible. So now it is not very difficult to imagine that if you could have vesicles which could enclose aqueous medium of small volumes, within that, if the primordial soup already had compounds that were contained, having these amino acids and other things, they would also be present. If there was sufficient amount present, and if there was a way in which these could react in that closed environment, then you could essentially start getting all this, components that would be fundamental to the growth or the presence of life. And that would lead to, if we made this jump, as we call a leap of faith, then we say that, well, there would be the first cells that could be formed from this kind of a primordial soup. So once that was done, our question now is, if that is done, and now we are here in this modern age where we are trying to understand the genetic code, is then, how did this all happen? So that through this history that we are trying to trace back, we understand, how this genetic code works. And we also begin to understand how this genetic code can be used, for example, in doing a lot of bioinformatics work, right? Or to understand, let's say, how genes function, or it can help us as systems biologists to be able to look at data that is there on the net and not get intimidated by it, because we have an understanding of how that data works, or what is the meaning of that data. So to understand that, I gave you this little part here where we talked about, how the genetic code was cracked. And we know that, the very elite group of Nobel laureates, 8 Nobel laureates, were sitting and trying to solve it. But it was solved by another relatively unknown person who had just recently joined a lab in NIH, and his name was Nirenberg. And how he solved it, along with his colleague Johan. And of course, the Nobel Prize was given to Harugovind Khurana and Hawley along with them. But the 3 people together had major contribution. One of them had discovered the tRNA. The other one had methods to synthesize precise sequences of mRNA, and the other person was able to devise experiments, right? So 3 of them together were able to crack the genetic code. Now, after that, post that, the reason why we are talking about this genetic code is to understand the structure, to understand why it happened the way it happened. Because if you do that little bit of retrospection on how the genetic code really evolved, then you have a better understanding of of data that you'll see later on. And we now see that there is a lot of research going on in this, but the most likely way this happened was that the most abundant amino acids first were selected by their hydrophobicity and structural properties to form into a category where G would have been the first letter, and the other one letter could have been used to define the other components. And we see that there are 5 of these top amino acids among the 10 were first coded for, and the rest came in later on, the next 5. So the top 10 amino acids, we saw how they evolved, and then gradually over a period of time, the other 10 were incorporated to give you a complete table of 20. Now in the table, you have what is called a start sequence, AUG, and your stop codons. There are 3 of them which are there. Okay, clear? Is this overview okay to you, everybody?
Yes, sir.
Huh? So now you have to get an idea of why we're doing all of this thing together. It's not without a purpose. It's with a reason why we're doing this, so that when we come to that stage where we need to understand these codes, we understand it. Okay, now evolution by itself is not only dependent upon just some kind of logical things that are happening, right? It's usually very there is also what is called game theory principles which come into play because of that we see so many variations in the biological world. Now, the reason why game theory becomes important is because for two reasons. First of all, it essentially will present situations which are not optimal, and whereas that solution is not something that is deniable or something that is not impossible to have. And that is the reason why you find that there's a lot of non-optimality in terms of certain things that are there in evolution. So, for example, this is the prisoner's dilemma and the snowdrift problem. So, two of them are there. But anyway, how many of you have seen this movie, Beautiful Minds?
Sir, I have not seen the movie, but I know that Prisoner's Dilemma.
Okay. Nobody's seen it, but then you need to see it. Okay, so there's a mathematician who has actually been responsible for this, really. So people from mathematics should be able to say, no?
Mind or Beautiful Minds?
Seen it.
Yeah.
John Nash, it was about John Nash.
Yeah, correct.
Yeah.
Yes, I've seen
Nash
that movie.
The Nash hypothesis, right?
Yes.
Yes, sir.
So essentially it means that, so that is how the prison dilemma works. So what it shows you is that, that there is, and actually, and this hypothesis, you can actually see it happening in real life also. Sometimes in meetings, if you're alert, you'll observe this happening in meetings and you'll know that, well, it's not going to work because, we're going to have the Nash equilibrium taking place. So this meeting is not going nowhere, especially in negotiations that take place. So what is the prisoner dilemma? So somebody can tell me from among those who have seen this movie. Anybody would like to tell me what the prisoner
Basically, it
dilemma
was
is?
about like two guys who committed a crime and they were put in separate cells. So, and like the police basically comes to each of them separately and asks them if you rat the other guy out, then you get a reduced sentence.
Yeah,
and
correct.
Like similarly for the other guy as well.
So what happens?
And if both of you don't accept to the crime, then like we'll let you go scot-free.
No, not that. So there are penalties which are different. So it's quite, I mean, what you said is right. So that is the way this whole problem arises in terms of description. So if somebody defects, that means if somebody says that his friend is the person who did the crime. He says, I'm free, I'm innocent, but my friend did it. Right? Or the other fellow did it. Then the other fellow so he's told, the prisoner is told that the other fellow will get higher punishment and you get a lighter punishment. And he tells it to the other fellow also. Right? Now, if both of them keep quiet and don't blame each other, then both of them actually will have the lower penalty. But it always happens that both of them get the high penalty because both of them tell each other, and so then tell on the other person, and so what happens is both of them get a harsher penalty. So that's usually what happens. So that's the prisoner's dilemma. So unless they have talked to each other beforehand, there's no way that they are going to. So the Nash theorem will be the solution to the problem. That it will happen, although there is a chance that both of them should be that they could have both had lighter penalties because by keeping quiet, they'll actually both of them will get heavier penalties because both of them are defecting. Okay. The same thing is the snowdrift problem, where two people are there coming from opposite sides and they're stuck in the snowdrift. Now somebody has to do is shovel the path and then both of them can pass through. But the question is, if they don't do it, both of them die in the cold. If one of them does it, he is paying the penalty of doing the extra work, whereas the other person is getting the benefit. So who will do it? So usually here it is the reverse because because of survival, somebody will take the penalty of doing the hard work so that both of them survive. Okay, so there's a reverse problem here. Clear? Now the question is how does all of this work out in the biological world? So this one, I mean, I'm not going to go through all of the details. So this one played a role mainly, let's say, in the utilization of metabolic pathways. Although they were energetically wasteful, that means there are redundant pathways which are there in cells. And you'll find this coming up time and again in bioreactors. So those who are working in the industry know that at certain pH changes, these microorganisms start becoming very wasteful. That means they consume the carbon sources that are being given to them, but they don't produce a product. So that is that most of the energy is getting wasted. This also shows in forests. Now the Amazon has been burned down, but that Amazon forest was one of the best examples. That you'll find that there are canopies of trees. That means all the trees grow to a certain height to get optimal to get the sunlight. So they'll all grow to an optimal height because you have to look at how much energy you're getting versus the energy that you spend to grow tall. So over a period of time, the seeds will start adapting and they'll go taller and taller, but all of them will all have a layer. At some level, they'll all stagnate and they'll form a canopy over the entire forest. So they'll all be at a certain height. And then you'll find one or two of these trees are going to be having extra height, but that gives them no really significant advantage because they lose by having to contribute more to maintaining that extra height, right? So that's the way this is. So we see that the question of non-optimality comes in, and this also we see in other aspects. For example, which animal has the best eyesight?
Owl.
Huh? Owl? No, owl is not on eyesight. Owl is not based on eyesight.
Is it eagle?
Eyes on sound.
Eagle.
Eagle has, yeah, eagle has one of the best eyesights. Does anybody know how they determined an eagle's eyesight that is better than human beings? How they were able to determine that? This is actually a very, very well conducted experiment. I'll come back to it a little bit. First, let me just talk about this. I don't know how much time we've got, 2 minutes, maybe more. So what happens is, so you look at non-optimality. So human beings have the best brain, but we are physically weaker, let's say the primates. Compared to gorilla of similar height and weight, will be much weaker. We are not the fastest creatures. We don't have the best eyesight, right? So there are so many other disadvantages we have as compared to other animals. So nature has allowed this non-optimality, and the principle by which this has happened is what is known as game theory. So, or one of the explanations that you could perhaps give to that happening is through game theory. Okay. So anyway, I'll end with this story, and then we can conclude this lecture. So the way that experiment was done for determining the eye of an eagle is the ability of an eagle to see, is that they trained an eagle to be able to fly from for a certain kind of for getting a food or a cue that was given to them, and they would go and get the so it'd be a piece of meat, maybe that would be something that would be a reward for the eagle. It is a trained eagle, of course, and then they had this huge hangar created where the eagle would fly from one point to the other to get the piece of meat that is there, or some kind of a reward that it would get in terms of food if it flew from one point to the other. So then what they did was they created a grid of a certain mesh size of black and white, and allowed the eagle to fly and to guess. So there would have been two different grids, and one would be of one shape, and the other would be of the other shape. So it would have to fly to the right one to get the meat, right? For example. So then they started making these grids finer and finer until the eagle could not distinguish which one was the right one, and they started making mistakes. Okay. So now, based on that distance that it could distinguish a grid of a certain mesh size, they were able to calculate the kind of resolution the eagle can have in terms of eyesight, and it's approximately more than 100 times that of human beings. So with that, I'll end the lecture today. Let me know if you have any comments. Anybody has any questions now? No? Yes, what? Somebody has to say something.
Not right
No,
now,
sir,
sir.
currently not.
No, sir.
Okay, but everything is clear so far, whatever I've done?
Yes, sir.
Gradually is going to
Yes,
pick
sir.
up now
Yes,
from,
sir.
yeah,
Yes, sir.
once these introductory lectures, yeah, okay. So once these introductory lectures are over, it's going to start picking up a bit more. So, all right. So when is our next class?
On Friday.
I think we have
Friday,
a
sir.
Friday is a holiday, no? I think
So I guess it would be
Then
on
on
Saturday.
Saturday, is it?
Yes, sir.
Yes, sir.
Yes.
Saturday will be working as Friday.
Okay, okay, all right. All right then. Okay. See you then. Bye.
Thank you, sir.
Thank you, sir.
Yes.
Thank you, sir.
Thank you,
Bye.
sir.
thank you, sir.
Thank you, sir.
Thank you, sir.
Thanks.